Hello, Dr. Cisse covering anatomy and physiology. Today we'll talk about DNA structure. It's part of chapter 4. On the left, you see chromosome. You see a chromosome with a pair of chromatid, sister chromatid. Then you see DNA here. Now, when you come to the bottom of this picture on your right, you see the same chromosome, but if I unwind the chromosome and I go into the detail of the chromosome, I will see the DNA. So in this video, I'll explain how this happens. Now, the questions we're gonna have in this video will be, what is the DNA? I'm sure you have heard of DNA. What is it? Where is it located? What is chromatin, nucleotide, gene, genome, sister chromatids? So what are these? Before I talk about genes, let's talk about necklaces and beads, right? So you have a necklace made from beads. So beads are pieces of a necklace. Now, if you have a necklace, you can have one piece of necklace, two pieces, and make bunch of necklaces, and then make different color of the same necklaces. That's how DNA works. It's like pieces of beads called nucleotides that you put together to form either one necklace, maybe two pieces of necklaces together, three, or it could be a bunch of necklaces. Now, when you have different group of necklaces, then you form chromosomes. Now, what is the difference between the necklace and the beads? The necklace is just an organized beads into a structure. It's the same thing. So nucleotides will be like pieces of beads that you put together to form DNA, and that DNA can be organized into chromosomes when the cell is dividing. Now, when the cell is not dividing, the beads are just like one long string of beads that is coiled in a very disorganized way. That long piece of beads together is called DNA. Now, it's going to be very confused at the beginning, but you're going to get it with time, right? At this point, all I want you to know, the beads are nucleotides. If you put a lot of nucleotides, you organize them into groups of necklaces. Each group is called a chromosome. And this, like purple, blue, red, in human, we have 23 pairs of chromosomes. So each piece here will be like a chromosome. One, two, three, and stuff. Now, so what is the DNA then? Now we know the nucleotide is each piece of these beads, right? But the whole long length, like the whole long necklace here, is the DNA. The long necklace here is the DNA. So now what is a chromosome then? If I take this necklace and organize in the specific shape, it'll be a chromosome. Now, why do you need that? Because when the cell is dividing, this DNA doesn't just want to randomly divide. It want to divide into organized way. So it organizes itself into chromosomes. So what about all the beads together in the human body? All the necklace together. Of course, there is the same thing, right? If I put all the beads together, I organize into necklaces, into group of necklaces. It's just an organized way from one piece to a group of pieces together. So all the necklaces will be the genome of the person. Like groups of necklaces will be chromosome. But if I take a piece of the chromosome, like a piece of the necklace here, that will be a gene. Of course, each piece of the bead is a nucleotide. So if you think about it, nucleotide will be small. You add many, many nucleotides together in a specific shape to form a necklace. That will be a chromosome. And a piece of that necklace is the gene. That's why sometimes you share a gene with people. You don't share a gene. It means you share pieces with people. Then the whole genome will be all the necklaces you have in the whole house, right? So where is the DNA located? It is in the nucleus. We talked about 4 different macromolecules: carbohydrates, lipids, nucleic acid, and proteins. Today we talk about nucleic acid because DNA and RNA are nucleic acids. And we talk about different parts of the cell. We say the cell membrane is the phospholipid bilayer. And then we have carbohydrate that extend from the cell. Inside the cell, we have organelles, mitochondria, Golgi apparatus, rough ER and smooth ER, lysosome, proteasomes, different things in the cell, and then the cytoskeleton. Of course, you have water, liquid inside the cell. That liquid is called cytosol. And then the liquid outside is called the extracellular fluid. So today we'll talk about DNA, which is inside the nucleus. So what is the DNA then? It stands for deoxyribonucleic acid. Of course, you see the word nucleic acid. So it is not protein, it is not lipid, it is not carbohydrates, it is nucleic acid. It is a long thread-like molecule with a uniform diameter but varied length, right? 46 DNA molecule, we call them chromosome. What makes it chromosome? Because they're organized into group of necklaces in the nucleus of most human cell. What is the average length? 2 inches long. What is the shape, the structure of the DNA? It is the double helix. It means it's twisted twice. It looks like a spiral staircase. Now, each side piece is a backbone of phosphate group alternating with deoxyribose. We'll explain what is this, right? So what is the nucleotide? It is the bead, right? Many nucleotide, it means polymers of nucleotide, just means many nucleotide together, right? And other nucleic acid form the DNA molecule. So we have stuff between, of course, the beads, but many beads, many nuclei together form the DNA. Now, what is this bead made of? What is the nucleotide made of? If you open this nucleotide, you will see that it's made of sugar, phosphate group, and nitrogenous base. Sugar, phosphate group, and nitrogenous base. That will be this bead. Okay? Now, I go inside the bead now. I see the sugar, which is deoxyribose here, the phosphate group, the POOO, you see all the O's, and then the nitrogenous base with the nitrogen. You see that it has nitrogen here. So the 3 pieces together form the nucleotide, which is the bead that come together to form the DNA. So this is a one base, nitrogenous base, adenine. And see, these adenine and purine, which is adenine and guanine, and pyrimidine, cytosine, thiamine, and uracil are the different type of nitrogenous bases that will make the DNA, and in the case of uracil, also the RNA, which is a type of nucleic acid. So in the nitrogenous base, what is holding them together? Hydrogen bonds. We talk about hydrogen bond in chapter 2. It is the bond. There's attraction between partially negative and partially positive part of the molecule. So the molecules of the nitrogenous bases are attractive to each other by hydrogen bond. So this hydrogen bond hold them together, makes them stay together. That's why the shape of the DNA parallel strands are uniform. It's the same length. It's the same distance between them because there's the same attraction between them. So in this case, what is the gene and what is the genome? A gene is a segment of DNA coding for the synthesis of a specific protein. It means a gene is a part of the DNA, is a piece of DNA that can make a protein. What about genome? All the genes of one person is called their genome. We have about 20,000 genes. Now, only about 2% of total DNA. It means there are part of the DNA that are not genes. Why? Because they don't code for anything. They're just between them helping. So the 98% not coding of the DNA are not called gene, because they don't code for something. They don't make something. They're just there to protect the other part. Now that we understand this, DNA, gene, and nucleotide. What is the DNA? A necklace. What is the nucleotide? Beads, right? What about a gene? It is the piece of the necklace that can make a protein. Now, why this is important? Because everything we're going to talk about in this lecture is how this gene makes something. How do you make stuff in your body? How that the gene is expressed in the body. Now, inside the nucleus, before the cell can divide, when the cell is not doing anything, the DNA are now well organized. They look like, for example, spaghetti in the sauce or some kind of cereal mixed up, right? The DNA is called then chromatin. Why? Because it's the fine filamentous, means like look like a thread-like DNA material complex with proteins. So it has all the DNA mixed off with other protein. Occurs as 46 chromosome in most cell, 6 feet long thread packed in a cell nucleus of 5 micrometer diameter. It means if the cell is not dividing, the cell is so slender, it cannot be seen with a light microscope. Only when the cell is dividing that we can see the very shape of the DNA that will be in a shape of chromosome. If you look at this picture, the bottom is like a chromosome, right? This is the chromosome at the bottom. If you unwind this chromosome, it will be like something you untwisted. When you go into the detail, that's when you see the DNA here with all the pieces of nucleotide, with all the adenine, thymine, guanine, cytosine connected by hydrogen bond at this level. What is histone? Histones are proteins that are crucial for DNA packing. It's like a piece of something, a material, a protein that can hold the DNA so the DNA or the DNA can be wind around it to be more organized. The nucleosome has core particle called the histone and then linker DNA, short segment of DNA connecting core particles. Now, why this is important? Because the DNA is so long, it has to be packed so tightly so that it won't take too much space in the nucleus. Inside the nucleus, the chromosomes have their own territory. That means each chromosome has a specific place they stay in so that they can be very well organized inside the nucleus. And genes can be turned on and turned off. What about sister chromatids? When preparing to divide, when the cell is dividing, the cell has to duplicate its genetic material. For that, the DNA has to be organized into chromosome, and each side of the chromosome is called the sister chromatid. Now let's look at the structure of the chromosome. You see the chromosome here. It is lots of DNA. You see the DNA things here rolling around here. Why this has to happen? Why do we need a chromosome in the first place? Because when the cell needs to divide, the DNA has to be organized such that each daughter cell can have their own DNA. So it has to be duplicated, replicated. It has to be double up so that the daughter cells can have each their own DNA. That's why the chromosome has to be organized this way so that it can give each daughter cell their own DNA. Now, if you look at this at the bottom, when you go up here, the DNA is here. You can roll it and roll it and roll it to make this structure. And this will only happen when the cell is dividing. Now, let's review the question. What is the DNA? What is DNA made of? Nucleotide. DNA is a nucleic acid, right? It's a nucleic acid. So where is it located? In the nucleus. What is chromatin? It is the structure of DNA when the cell is not dividing, when things are not organized. What is the nucleotide? In our case, it's like the bead. It's the piece, right? So many nucleotides makes what? The DNA. What is the gene? It is the piece of the DNA that can code for a protein, that can make something. Remember, only 2% can make proteins. What about the genome? All the genome of the person. What are sister chromatid? It's just the pieces of the chromosome that when the cell divide, each one will become their own DNA, right? All right, so I'll see you in class. Thank you.